to my channel Study with me PCMB. This part of molecular basis of inheritance will learn about the discovery of DNA, how DNA was discovered. So let's start. So a scientist, Friedrich Miescher, in 1869 observed an acidic substance, in nucleus. So in 1869, Friedrich Miescher observed observed a presence of an acidic substance in nucleus. He named that substance as nuclei. Because it was present in the nucleus. So you can see here it was in 1869. So at that time, due to some technical problems or lack of technology, there won't be a proper microscope or microscopic studies was not done in that time. So it was kept like that for a long period of time. So let's see then what happened. Before that, we can study about two scientists. Watch this video till the end because it is very important for your board exam as well as for NEET. So those are Maurice Wilkins and Rosalind Franklin. So this Wilkins and Franklin's proposed X-ray diffraction data. They made this X-ray diffraction data. So remember these two scientists' name and what did they done. So let's come back to our DNA, case of discovering DNA. So after that, in 1953, around 100 years, from 1869, Miescher first observed an acidic substance in nucleus. He named it nuclei at that time. Then after many, around 100 years, in 1953, James Watson and Francis Crick proposed a simple but famous double helix model for the structure of DNA. Double helix model of DNA. So these are the two scientists and this is the double helix structure. It is nothing but the symbol, a 3 dimension of the normal DNA. Like this. It is in a plane now, but that 3 dimensional. They observed it. They created a structure first. Then after that, we will study what are the hallmarks. The one of the hallmark was the base pairing between two strands of polynucleotide chain. Watson's and Crick's double helix model hallmarks was base pairing between two strands of polynucleotide chain. We have learned about polynucleotide, dinucleotide, all these things and how are they formed. So, the hallmark of this double helix structure was the base pairing. It was base pairing. You know what is base pairing? That means this way. And then what is, you may have doubt what is the strain. This is the structure. We can draw the structure like this. This is base pairing. Just imagine it as a DNA, and you can see that there are two polynucleotide chains. That is, one is in this way and the next one is in this way. So this is one polynucleotide chain. This one is the other polynucleotide chain. This is base pairing. That's all it is. The hallmark was base pairing between two strands of the polynucleotide chain. This is a polynucleotide chain. This is also a polynucleotide chain. And there are base pairing. There are base pairing between these two polynucleotide chain. So this much is there in this discovery. After that, we have to learn about a rule proposed by Chargaff. Chargaff proposed the rule of base pair. So let's see what are those rules. There are two rules. First one is we have learned about purines and pyrimids also. Purines pair with pyrimids. Purines pair with pyrimids. The rule is saying that purines pair with pyrimids. And I have also told you that there are two types of purines and there are three types of purines. But now we are talking about DNA, not RNA. So what will be? Uracil will not be there under pyrimids. So if you are elaborating the purines, that means adenine. This is a purine. Adenine pairs with thymine. Adenine pairs with thymine the same way like guanine. Guanine pairs with cytosine. Guanine pairs with cytosine. So this is the pairing. It is very important. And one more thing I will tell you, it is not in our textbook. In this part, I am taking two paragraphs today. That is in adenine. I am representing adenine as capital letter A and thymine T. This is the pairing. And guanine is paired with the cytosine G and T. Here will be 2 hydrogen bond. Here will be 3 hydrogen bond. Adenine and thymine, between adenine and thymine there are 2 hydrogen bond and 3 hydrogen bond between guanine and cytosine. This is thick, which is very important also. So this is the first rule. The first rule says like this. Then you can move to the second rule. What is the second rule? says that the quantity of purine is always equal to that of pyrimidine. This is purine and here is pyrimidine. Now, for your better understanding, I am writing here purine is, suppose consider purine is of 20 gram. Just an example. Or just consider it is of 20 laddus. Like that, anything you can consider. Suppose purine is 20 grams. And pyrimidines, if you check the pyrimidines, it will be 19.9 grams. Like that, you can know. If suppose purine is, and then pyrimidines is 35 gram. Pyrimidines is 35.1 gram. Like that, there can be slight variation. But the rule says the quantity of pyrimidines is always equal to that of pyrimidine. The quantity of purine is always equal to that of pyrimidines. So this rule states that. And further, in this second rule itself, it is stated that the ratio between adenine and thymine and guanine and cytosine are constant and equal to 1. The ratio between adenine and thymine pair, they have 2 hydrogen bonds, and with the cytosine, with the guanosine, guanine and cytosine pair, they have triple hydrogen bond. The ratio between adenine and thymine and that of guanine and cytosine are constant and always equal to 1. This is what taught. I'll repeat the statement once more. The ratio between adenine and thymine and guanine and cytosine are constant and equal to 1. So, related to this concept, there may be questions. So, let's see one question similar to this topic. So, I am not writing the question here. The question is, if a double-stranded DNA has 30% of adenine, and calculate the percentage of cytosine. In the DNA. The question is in the DNA, in a double-stranded DNA, adenine is of 30%. Okay, you have to calculate the percentage of cytosine present in that DNA. So, you have learned how are they pairing. So, if adenine is 30%, then automatically thymine, that is this pyrimidine, will be 30%. That means purine and pyrimidine, the quantity of purine and the quantity of pyrimidine are always equal. So here are the bonding. Adenine and thymine are getting bonded. So what the quantity of adenine is equal to the quantity of thymine also. Then if it is in percentage, so percentage means it is out of 100. So 30 plus 30, 30 plus 30, 30% plus 30%. The answer will be 60%. Then we can see the next rest. So here is 60%. So 100 minus 60, what will the answer? It will be 40. So rest there are 40%. Then we know about guanine and cytosine. Guanine and cytosine pair with each other. So the quantity of guanine is always equal to the quantity of cytosine. We have 40% with us. both have to get equal. So what will we do? We will divide 40 by 2. So the answer will be guanine will be of 20%. Cytosine will be of 20%. This is our answer. We have to find the percentage of cytosine. That was the question which was given. So this way we can find that. I'll repeat it once more. So the percentage of adenine is given. Whatever the percentage of adenine, or in what terms the percentage of adenine is given, that will be equal to that of thymine, the quantity of thymine. And the rest is guanine and cytosine. We will get the value of that. By adding, we will get the value. Then after, in total, which is total, it is of 100 because it is of a percentage form. So the rest is of 40%. So this should be also the guanine and cytosine. The quantity of guanine and cytosine must be equal. So the 40 will be divided. 20 will be here, 20 will go there. So total there are 40, here 60, 100. This done. This is a question. This type of questions you can accept for exams. Then we can move. The base pairing is a very unique property to the polynucleotide chain. Base pairing is a very unique property to the polynucleotide chain. They are said to be complementary to each other. Means this consider this as a DNA. Along, it is not in that shape, it is elongated. Here is the base pair. I will tell you one more thing. If here adenine is percent, then here consider is cytosine. Then here is guanine. Here thymine. Then adenine, cytosine, guanine, etc. This I am just writing it as a refer for your understanding. So we know that there are two polynucleotide chains. This is one polynucleotide chain. This is another polynucleotide chain. So we know the sequence of one polynucleotide chain. So then we can simply find the sequence of the other polynucleotide chain. How? Adenine always pairs with thymine. Cytosine always pairs with guanine. Guanine always pairs with cytosine. Thymine always pairs with adenine. Adenine always pairs with thymine. Cytosine always pairs with guanine. Guanine always pairs with cytosine. This will be the sequence. That means they are said to be complementary to each other. If the sequence of bases of one stated is known, then the sequence of the other strand can be predicted. We can predict the sequence of the other polynucleotide chain or the other strand. DNA is of double stranded. It is of double stranded. If each strand from a DNA acts as a template for a synthesis of a new strand, the two double-stranded DNA thus produced will be identical to the parental DNA molecule. Because of this, the genetic implication of the structure of DNA become very clear. This is our last concept. That means if you are taking a parent, I can call studied about amoeba, parent amoeba, and how it is become to daughter amoebas. Like this, just consider a DNA, a parent DNA. Each strand from a parent DNA acts as a template for synthesis of a new strand. The new strand is our daughter DNA of two double-stranded DNA. That daughter DNA will be identical to the parental DNA molecule. Because of this, the genetic implication. Genetic implication genetic implication of the structure of DNA becomes very clear. So like this we can easily find. We can easily implicate the structure of DNA. So if you need the handwritten notes of this chapter, just post a comment below about the thing that you need the notes of this chapter. So in this part we have learned about some scientists, their contribution. About 5 scientists we have learned, and who found the DNA, and how is their pair, how many pair are there, hydrogen bond case, hydrogen bonding. Then after that about DNA, how the sequence is getting there, and how we can find the other sequence of the other strand if one of the strand is not. Then after that, how it is the genetic implication of DNA, how it is very clear, etc., etc. we learned in this part. So thank you.